So good morning, everyone. I will show you today what I did in a previous work about the red blood cell content and how I set up a database about the proteins involved in this red blood cell environment. And this database is called Respire, the repository of enhanced structures of protein involved in the red blood cell environment. So this is a work I did in Paris, here, with my colleagues at the DESIM laboratory. So it's a laboratory specialized in membrane modeling of virtual experiments and virtual modeling of membranes and of protein membranes. So this work was done under the supervision of Catherine Echebest, and this is a work which is published now in PLOS ONE. I will show you later what we did on that. So the first thing you have to remember is that when you are talking about a cell, it's a very complex entity, and it's made up of many proteins, many compartments, many DNA and proteic entities, and of course of many metabolites. So when you take this into account, it means that you have to take the full complexity of a cell to try to understand how it works. So the first thing that you have to take into account is what's in a cell and what's the cell that you want to study. So I gave you here all the things that we call red blood cells, and not only red blood cells, sorry. So you can go on the link, provided link here. I will give you the presentation just after. So you can check what all these cells look like and what they contain, but you see from their shape and their size that they're very different. So they are all called blood cells. So you find them in our blood. So they are circulating all throughout our body, but they are pertaining to many different functions. First, you get the monocytes and lymphocytes, which will be involved in the immune system. Then you get the neutrophil, eosinophil, and basophil, which also will be involved in the recognition and degradation of external entities, viruses, bacteria, or other cells coming from other organisms. And so the top cells here contain a lot of differentiation factors to attack stranger cells or stranger components of the body. The macrophage also is very important for the first line of our immunity. And the two last cells here are the one that we talked about, and especially the erythrocyte. And this is very well known because this cell is the cell giving our blood its color. It's the red blood cell, it's the erythrocyte, and it's void of organelles. If you compare it to the other cells there, you will see that there is nothing inside. It's completely flat. And it's red because it contains a lot of iron, and this iron is coming from hemoglobin. And then the last component of the red blood of the blood cells is the platelets that will aggregate when you have an injury on your body. So it will aggregate to prevent blood release. So when you go to the detail of the red blood cell, so the erythrocyte, you see that from the last step of differentiation that you see here, so it's slightly less red color than the one before, but you see it's the same shape and the same organization. There are many different events that happen to give this cell. So first, you will start from a germinal cell. So this germinal cell is called an hematopoietic stem cell, and it needs a different steps to activate, to translate from the cell, the undifferentiated cell, into the red blood cell that you all know. What happens? So first, we can see here the bone-forming unit pre-erythrocyte, which is the cell that you will find in the bone marrow. So inside your bones, you get a reserve of cells, and they will be there and produce a very high rate in the bone marrow to provide the red blood cells. So at this stage, you cannot differentiate easily these cells because they present the same marker, which is CD34, so cluster of differentiation 34. Then, upon the expression of different factors and cytokines, you will see these cells will begin to transform in colony-forming unit cells in the erythroblastic lineage, then into basophilic cells. And here you start to see very important receptors, so the c-KIT receptor, which is a tyrosine kinase involved in many cellular process activation, but also sometimes very important in cancer cells, where it is upregulated or completely deregulated. And you start to see a famous receptor, which is the EPO receptor, so the erythropoietin receptor, which is an element where you will enhance a lot the quantity of red blood cells if you increase the amount of erythropoietin in the body, and it's well known in sickling, for instance. So you have already the appearance of this receptor at this period, but the cell is not completely void of organelles. And I remember you that the erythrocyte is void of organelles. So you need to continue the differentiation, and then you see the appearance of other differentiation factors, so CD71 or 36. And these different factors are important for FACS analysis, for differentiating the cells. So you have the shape and the appearance that you can measure using the colors like this, histochemistry, but you don't do that all the time. You won't take living cells and color them. You have to fix them to obtain this. So if you want to filter, to find which cell corresponds to which one, you have to take these markers into account. And the last one, which start to be very specific of the red blood cell here, is when you will start from a basophilic to a polychromatic to acidophilic to reticulocyte to red blood cell. You see there are two markers here, which is the glycophorin A and band 3 proteins, which are very specific to red blood cell. And you see from the shape here that are shown that from a big cell with a large nucleus, so close to the one that you have in the other cells, you see here, you have the limits of the cell and then the nucleus, which is quite huge. Then you will start to expel these organelles, and you will split the cell into a reticulocyte, which contains all the remains of the cells, and then the reticulocytes, which itself is able to perform the main function of the reticulocyte, which is transporting oxygen through hemoglobin binding, oxygen binding by hemoglobin. So this process is very important. As you see, there are many markers, and this is performed every day on our cells at a very high rate. We have millions and millions of cells produced every day for getting the final erythrocyte. And I remind you, because you have probably heard of that already, that the red blood cells has something very specific, because this cell will last about 100 days. So this cell is very specific, specialized, and has a long lifetime, and is expressed at a very high rate on our bodies. The most expressed cell or differentiating cell in our body compared to all the others. You don't do muscular cells, for instance, at this rate. It's impossible. So when you go into the detail, because this is what is important, is why do we qualify these compounds with large names, and what are they providing to us to understand the function of these red blood cells? So even in here, you find it completely void of organelles. In fact, it's still very complex. At the surface of the red blood cell, you will see a detail of the complex still existing when all the process has been performed. So the name that you that we assign to these complexes is coming from all this story. So band 3 here that you see means that on a blot, you had the migration of cells of proteins after digestion, enzymatic digestion and expression, and they were moving on the blot at a speed which allowed them to be the third band on this blot. So you had 1, 2, 3, 4, and more. You see that. So the at that time we were not able to see which proteins were pertaining to this, but we will show that there are band 3 complex, and you see it's made of many compounds, but also the 4.2. Here, there's also 4.1, and of course, 2, 1, etc., many, up to 7, 8. So this is the assembly of the current knowledge on the surface of the red blood cell. And you see here the lipids, which are indicated by a circle and tails. And inside this lipid membrane, you have proteins embedded in it and names that you may recognize. So I will give you some hints about them. So first of all, these proteins contains most of the time an intracellular part, a membrane part, an intracellular part, and an extracellular part. And you see here a lot of glycans. So the proteins are very important, and in context of the COVID-19 pandemic, we know that these carbohydrates are very, very important for the spike protein, for instance, to recognize our receptor. If you remove the glycans, the epidemic will be very, very different than what it is at the moment. So this defines our self. So why do we differ each one from each other's from an immunology point of view? It's because we have different glycans, and we have also different mutations or variations on proteins. But when you take a look at the assembly, you will recognize here what is known as RH, so the rhesus, or Lewis here. And in this situation, you see there's no glycans. So these proteins, which are present at the cell membrane, are very important in transfusion, because when you want to transfer the blood to someone because he has a bleeding issue, so you need to give him transfusion, you have to be compatible with his blood, otherwise you will kill this person. So the reason why you will kill this person is because there are variation on the RH, for instance, so the rhesus, and you know, it's you have it or you're not, it's plus or minus. But there are many other red blood cell antigens. And if you look at the protein by itself alone, it's very difficult to understand, but if you see the complex here, you see the difference between the different all the partners and how they are arranged. So you see the Lewis here, which is probably a red blood cell antigen, which is less common, and many of the proteins here are known to be involved in the recognition. So you see, for instance, the Duffy antigen, the Kinase antigen, and again, they are part of a larger complex, also bound to the band 3 complex. So to sum up, the membrane context is very important, and how this context is completely linked to the red blood cell architecture. It's because you have spectrin here, which is linking all these complexes. So it's very difficult to see if all these proteins are assembled in the same way or split like this. We have no idea about that. We have just indirect indications about that. But the chains that you see here are the assembly of what we are knowing about that. So the red blood cell is, so the hematopoietic cells are very diverse. If you take a look at one cell, which is very important, the red blood cell, it contains a lot of proteins already at its membrane surface. So what was the question? The question was, how many proteins do we have, and are we able to model them? So when you start these kind of questions, people always say it's not useful, or we know already everything, or we don't see the point, or yes, it's very, very important. Please do that and give it accessible to us. Of course, depending on the context, the person who will look at the database, you will have different expectations, but we know from the existing databases what we can determine, what we can derive. So first, we can make use of existing databases. For instance, UniProt, the NCBI, they contain a lot of information which are entry-driven. So it's not a complex that I showed you, but it's protein by protein, gene by gene, you have a lot of information. They are also made up of combination of information and links, and this is what makes them difficult to use. We see that on the bioinformatics session on course. It's very difficult to see on which database is important for getting one information, and if this database is the reference database for this information, or if this information coming from another one and linked into the card that you are seeing. So it's most of the time it's an assembly of many different information assembled in one card. And if you want to have the detail, you have to click on one link to go on the other database. So this is known, established, but complex to use. You cannot do complex requests. So what are all the red blood cells antigen known on the erythrocyte? So this is one example. Most of the time, if not all the time, they don't contain the structural information. So you don't know if here it's only a cartoon or if you have the real 3D structure of this system. And as I showed you here, it's very important to understand what are the details of these proteins, because many of these proteins are involved in disease or in cell malfunction or non-malfunction. For instance, here the Duffy antigen is known to be involved in the infection of the malaria parasite. So if you get a mutation here, you may be protected by against this infection. So this is a major point. It's very specialized and it's difficult to get that. And most of the time also it doesn't provide the functional annotation. So you will get what the gene is for, but not how this gene is involved in a network of interactions. So there were already a lot of databases on red blood cells to address these needs. For instance, the Rhesus database, one database which was BGMUT, but it was only targeting the alleles found for many different proteins, and it's now very discontinued. It's discontinued for nearly 10 years. Most of these databases are not updated regularly, so they are set up once because someone did a lot of work to set it up. Then the person is not able to continue the work on that, so he has to. So the database is now no more updated and sometimes completely removed. And sometimes you get information which is private. So there are well established in the lab facts about a protein, about a gene, etc., and they are not available. So this is why we have started to set up a database addressing all these issues and more. So what is Respire? So the first thing was to see what's in the red blood cell environment. So this is really the perspective of this work, and it was also focused on the terminal stage of the erythrocyte. So experimentally, it's difficult to determine if you get data from the reticulocyte, so the one containing organelles which are split from the red blood cell, the erythrocyte by itself. So this is why sometimes you get confusion about that, but this is a technical issue. The interest of Respire is really to be cell-centric. There are no databases which are from one cell able to provide all the information that we know actually on this cell. So you get many, many predictions, you get many, many data in the the literature, but once one stop shop for getting this information was not available at that time. We know in the opposite that there are red blood cells antigens. They are characterized. There's the community naming and listing them. I performed direct queries about that. I will show you them just after. And there are complex queries that can be performed. Since the membranous context is complex, because I showed you transmembrane proteins, so they are mostly involved in the membrane. But many proteins are just anchored to the membrane, or just one helix is traversing the membrane. So it's very difficult to get a broad definition and a consensus definition of all these categories. So I've provided two ways that are well established, and they are not always providing exactly the same results because of these differences in definitions. So what was done in Respire? So the first thing is, since we want to focus on proteins, the information, the main information, the name, the sequence, the length of the sequence, the molecular weight, and many different data are available from UniProt. So they are taken as them. The information are really extracted from UniProt, and this update is performed monthly. So each month, so it has changed from this the diagram, because now UniProt has changed its release schedule. Is more now than 2 months than 1 month. But following UniProt update, there's an update of Respire. What we do, what is the information that you integrate above UniProt? We take the data from the experimental structures delivered in the protein data bank. I will show you why, but it's very difficult to produce a reliable model of red blood cells. So if you want red blood cells proteins, so if you want to give the information, you have to take what is existing at a certain time. So you have to redo it very rapidly, monthly in that case also. The information which is really lacking most of the time on many databases is a very comprehensive description of pathologies associated with one protein. So you will get very broad description, for instance, transfusion problem, transfusion trouble, but not the detail of what can happen, because sometimes when you the transfusion issue can be linked to one mutation and not to the whole protein, for instance. So you don't get this kind of details. When you want to get all the interactions, you can take the information from BioGrid. So BioGrid is assembling a lot of experimental information into one database. It mixes a lot of different information, so you should have some care when you look at the information, but it provides a way to see the network of interacting proteins, and I will show you that. It's very interesting. One protein is most of the time not only made of one domain, one function, for instance, an enzyme, but it will contain most of the time one domain, one functional domain, then another one for another function. If the protein is very big, you will get many different domains. Most of the time, you get at least 1 or 2 different domains, and so they are computed using Interpro. And the last one is really how we can change our view on how to organize data, and it has been done nearly more than 20 years ago now. It's the gene ontology. So how can you say that the protein is involved in the pathway? How can you say that gene is important for a big process like cell differentiation? This is the gene ontology consortium that set up those rules. So they define the names, they define the interactions and the directions. So you get a hierarchical overview of the genes. And this is really something important for the annotation of our genomes, because other than that, it would be very, very difficult to assemble all the data. And so this is still an ongoing work, but many processes are very well established. And with this work, we were able to see that the core principles of the cell organizations are very conserved among a lot of organisms. And so we are much closer to a worm or to a plant that we were expecting at the beginning, and in particular what we were expecting from the visual inspection. Of course, we have not the shape of a worm. So the takeaway message here is Respire is always up to date, or at least aims at being always up to date. For instance, the work done in these databases is to standardize names. So there are common names that were used for maybe 20 years, 100 years, but at a certain point, there's a requirement to set up a common name for all, not only on one species, but for all the species that we know that we have the genome. And one example here is the Duffy Antigen that I showed you already here. So it's well established that it's the Duffy Antigen. Nobody will change that. And the Duffy Antigen, they have decided to say, okay, it's an atypical chemokine because the real function is a chemokine, so a sensor of chemical entities. After that, so here are examples of what you will find in the database. First, we have a lot of information coming from sequence. So here you have an extract of what is a sequence with color-coded conservation. So to get this information, there's a lot of processing. You see, it's indicated here. It means that it will take you 16 days on one CPU to produce this image. And you can compute the conservation. So you see the color code is from no conservation to high conservation. So the more red, the more it is conserved. And so you see graphically on the length of the sequence if there are regions which are more conserved than others. Okay. And of course, since it's a lot of computation time, it's provided also as download buttons that you can click to get the information. So you have the result of the calculation, which is used to produce this information. And like this, you can do your own analysis on your computer if you want to continue with that. Then I show you here that for one protein, so the protein code here is an aquaporin 1, which is described here. So the UniProt code is P291972. And you see that this protein is made up of a big domain, which is so the large description of the domain, but Interpro is able to define smaller domains. And to produce this graph, it will require about 4 hours for one protein. If you click on a domain, you will be able to grab all the proteins getting that on Respire. So I will show you an example after the screenshot how it works. And you see there are also some gene ontology predictions, which are split into 3 categories: biological process, molecular function, and cellular component. And you see that this protein, for instance, is important in transport of water because it's aquaporin. There's an activity, so this transporter needs energy to transport water. It's not passive. And then it's a membrane compound, so it's located in the membrane of the cell. When you go to the structure tab, you will get now all the templates that are available for a given protein. And you see here, there are pre-computed cartoon to display buttons to display cartoon information on the structure of the protein. So you can click here on the right to select what is known on this protein. So in that case, it's a protein which is well annotated. So we get a lot of information. You can see the helices in red, the sheets in yellow, and the coils in white. So you have the complete shape. It's interactive. I will show you that. When you look around the protein, you can see the details, and you can select here amino acids to display the location on the protein. So it's already quite interesting in this interface. If you want to do more, you can download the file, and then you can again use your own software to see that. This small window here indicates also if the location of a mutation is supposed to be with no consequence or with high consequences. And if you click on that, you will get more information about the details of this prediction of this information. So this part is, again, you get the result, so you can see that. But to compute that, this information, it's at least 1 day of calculation or 6 hours, 6 CPU days for computing that, but up to 100. It can take 1 week on a cluster to predict this kind of information for one protein. So why is it very difficult to assemble this information? Why is it taking so long? It's taking so long because we need to have the 3D structures of these proteins, and this is the biggest problem in biology for years, and we still are struggling to find a way to get that easily. The first thing is it's bound to the experimental data available, and this experimental data is coming from experimental work, from X-ray crystallography, from NMR studies, from the recent advances in electron microscopy. For instance, it was very important this year with the release of many proteins with electron microscopy. If it was only coming from X-ray crystallography, we would have not so many crystal structure and microscopic electron structures of the spike proteins, for instance. And you understand that it's very important to map the amino acids mutated for the variants. And so when you don't have the crystallographic structures or the NMR or the EM structures from the PDB, then you have to predict them. And when you start to predict, you see you have less data. And of course, if you have less data and you're trying to do a modeling procedure, then it's less reliable. You see, not as reliable that when you start from the experimental data. And in the base cases, I mean, you don't need to predict the structures. You get the information already, which is the case that I show you here, which is I would say the base scenario case. When you have even less information than what is required, so you get really a part of the protein and not a lot of them, you will consider that the protein is made up of different blocks that you will assemble all together to predict the shape of the final product. And in that case, the reliability still becomes lower. And one of the methods known to be quite performing quite well in this situation is iTester, which is an iterative process where you will get independent blocks of proteins, and you will try to assemble them in 3D. And in that case, since you have many different compounds, I was searching about, okay, for instance, if you have 2 shapes, there are many ways that they can interact. They can go inside it, they can go aside, they can turn on the other side. So there are many degrees of liberty. So it starts to be very complicated. It takes a lot of time, computing time, and the reliability is very cautious. So you have the information, well, you have a model after that, but you have to pay very attention. And in the end, when you have a lot of, you have really a paucity of data, nearly no data. In that case, you will do a de novo or ab initio modeling. And this is the most complex task because you will consider nearly proteins as made up of tiny blocks, like Lego blocks, of a bunch of amino acids, so 2, 3, 4, 5 amino acids. So just small parts, and you will try to assemble them by constraining their shape and their organization. So it's even more complex than the step before, and it means that it's even less reliable. So there are cases where it works quite well, but sometimes you will get very bad models. I will show you some of them. So the this stage is really the core work that was done on Respire is predicting the 3D structures of all the proteins in the database, and I will show you there are a big amount of proteins inside the database. And the first case is only 10% probably of the proteins. The second one is another 20%, so you get about 1/3, which is quite reliable models or existing proteins, so you don't need the model. And then it means that more than half of the proteins are models that you should take care really carefully about because we know from the design of the experiments that they contain information, but they contain also a lot of errors. So you have to be very careful about the usage. So here are 3 examples that you will find on the database. So for instance, the basicin. Basicin is one of the protein involved in the red blood cell antigens definitions. And you see here, for instance, 2 positions which are known to alter the phenotype of the antibody recognition, and you see why. Here's the basicin shape, and these 2 positions are exposed to the solvent. So this is why it will change the recognition by antibodies, for instance. Here, it's a MAP kinase. So MAP kinase, you see here the process of assembling different models, different shapes into one, because there are parts of the model which are quite well defined. You see here beta strands. You can see here helices. Some of them are curved, and they are coming probably from the template, from the structures they are made of. And there are many uncertainties in other parts of the protein. You see spaghetti like here or here. So it's not well defined. It can be that this structure is not defined. This happens. There are many parts in our proteins which are not clearly regular. We call that intrinsically disordered proteins. But most of the time, what happens is that the model is not correct here. Okay, so we have you have to take this model to a starting one, and you need to refine it, and it's a lot of work to get a better model than that if you have this kind of situation. And then here, it's a protein which is the BCL2 modifying factor, which was performed using Rosita, so the highest difficulty. And in that case, you see that again there are regions which are well defined and other regions which are not. And I know well this protein now, not at the time that I produced the model. And I know that one specific part of this protein is known to be intrinsically disordered. So it's logical here to have no structure here, and it's made up of helices. So the model is mostly correct here. There's probably not a beta sheet here. So this model should be updated. But in some part of the model here, it's known to be not structured. So this model contains valuable information about some of the parts. And one of the missing part here is this protein is one of the membrane proteins that I showed you. So one helix here, probably the orange one, is inserted into the membrane. So you have the core of BCL, which is outside the membrane, but one helix is inside it. Continuing into the tabs that you will find in Respire, you will get a lot of information about the interactions. So this is not very long to compute because you have to take the data from the BioGrid website that I showed you, the BioGrid database, and you can see the direct interaction on from this protein to the other one. So when you hover on these pills here, you will get the definition. I will show you on the interactive part just after. But it shows you the network of interactions, and it's interactive. So you can drag the pills to change the display, and when you hover on it, you will get the information. So it's important to get the name, and only the UniProt code is shown there because the name will be too long for the pill. It will be unreadable. I told you a very tremendous amount of work was done in the end of the 90s and early 2000 to assemble our knowledge into logical pathways, processes, components. And this is what you find for any any information in database. So they are assembled into three main categories. I showed you them already, but it's more detailed here. They are assembled into the biological process, so where is it involved, into the cellular component, so where is it present in our cell, and you see it's very different, I will show you there, and what is the function of this protein. Well, and you see here that we always resume or simplify the function of one protein to one specific mechanism, but you see here in this screenshot that you get a lot of information, and it means that every protein, every gene is involved in many, many different processes. For instance, here, this protein is involved with ammonium transport, or carbon dioxide transport, or the cellular homeostasis. So you see, this is a very broad process, the cellular homeostasis. So it means it will help in keeping our body to 37 degrees, for instance. The components, so it's much more than the big components that you know, so the cytoplasm, the mitochondrion, the reticulum endoplasmic, or the nucleus. We get much more detail about that. So for instance, at the edge of the cell, inside the cell, and sometimes you will get very specific proteins known to be involved in other compartments than the one found in the red blood cell, for instance, in spermatozoid. And the molecular function, so again, you will see that they are involved in transporting the ammonium. You see the difference here. The biological process is ammonium transmembrane transport, and the activity is added for the function. You will get, for instance, protein binding, which is a very broad keyword. And the function here of this website is when you click on the link here, you will find all the proteins where this mechanism has been found inside the proteins of the red blood cell. And when you click on a small image here, you will go to the upstream information, which is available in the genome from the Gene Ontology Consortium. So again, this is very important. So this is why I gave you a detailed picture of the information. What is gene information? It's an up-to-date, comprehensive computational model of biological systems. So it's much more than definitions. It's really a model of all the interactions, and it's made for computers. So it contains a lot of information to arrange the knowledge into a graph, into a complete vision of our knowledge, with not only the definitions, not only the interactions, but also the hierarchical relationships. It has been set up from the work done after the sequencing of the budding yeast, the nematode worm, and the fruit fly Drosophila melanogaster. And you see that it was nearly concomitant with Arabidopsis thaliana, so the plant, and Mus musculus, and of course, ourselves. So it was really within 10 years time frame that they have assembled that. And surprisingly, they found that many processes were common. So ammonium transport is the same in many organisms, not only in human, in probably the same in the nematode world, for instance. So you can have a look at this article. It will be it's very interesting to see. It's from 2000. You see, this is where they describe how they have organized these relationships, but it's very important. From the genetic point of view, when you have you want to study the shape and architecture of a gene, I'm taking this information from OMIM. So OMIM is the online Mendelian inheritance in men. And it's very interesting. Why? Because it's a human that wrote that. It's not derived from a computer, but really a human has wrote this information. It means a human has read this paper and wrote this sentence. You see, they describe, for instance, how it was found, what was done to describe this information. And since OMIM is doing a lot of information, a curated amount of information, I'm not providing all the page here because I don't want to hijack their content. OMIM needs funding. So when you need more information, you have to click here to go to their website, so you get the complete detail of one sentence, but you have already the beginning of that. So in that case, it means this gene is not involved in a pathology, but I will show you cases where it's the case. I'm not going into history and links. I will show you them on the database, but the information about that is quite clear. History is what happened on this card, if there is a revision, etc. And the links is about the external links that you can consult to get additional information from the database. So if you take into the database numbers, you get nearly 2,500 proteins listed. So each number that I gave you, the 1 hour calculation or the 100 hours calculation on one CPU, you have to multiply that by the number provided here. So it gives you an overview of the very high computing time it took to provide this. There are many, many variants from each protein. On average, about 4 allelic variants which are known. So it means for every gene, there are at least 4 alleles, and probably much more than that. There are nearly 10,000 gene ontologies for describing only on the red blood cells the proteins and the interactions that describe the network. Many, many different binding partners. So there are partners which are involved in the red blood cells, so they're really binding, for instance, like in the bond 3, bond 4. But some proteins are known to interact with other proteins in other cells, for instance. Many different references coming from all this data are also indicated in Respire. You can have a look at that. Most of them are coming from the 3D structures where this information is available. And from what was possible to do at that time and what can be updated, it will be probably updating this year or the year after, there are more than 1,400 proteins which have a well-defined structure. So in theory, when you look at that, there's 1,000 proteins missing, but it's impossible to predict them with the current knowledge. Okay, we know that it's lacking from the existing knowledge. When you browse into the databases, and even if you go to very tiny details, we know that we have no structure. So we have no idea about that, and it's a well fact established. Even for humans, we know that about half of our proteins, we have no structural information about that. This is called a dark proteome. And even if we have some information, there are some proteins for which about 10–20% we have no idea of what they are doing. So we have no function for these proteins. So the knowledge is really not completely established, and we have still some work to do to continue that. So it's up to you. So I try to produce as much possible as models, but for different parameters, it was not possible to produce all the proteins. So there are 300 proteins lacking, but it's impossible because they are too big or too complex, or the methods fail. They are completely irrelevant. And if you want to do very precise models of one protein, then you have to give a very specific attention and publish that. And only 3 on all these proteins were performed by my colleagues and published. So 3 are available also from this website. So again, this work was performed there, and I will show you now a demonstration of that.